Okay, welcome back. Let's continue our discussion in module 8. So today we are going to look at the problem of multiple sequence alignment in the context of pangenome graphs. In general, multiple sequence alignment is a fundamental problem in bioinformatics, but we are specifically looking at this problem in the context of pangenome graph construction. So if you remember from the last lecture, we discussed various graph based representations for representing a collection of genomes. So for example, one definition we looked at in the last lecture was this pangenome graph, where we said that this is a directed graph in which the nodes of the graph are labeled with sequences and edges imply adjacency in a genome. So at the bottom you see a toy example of a pangenome graph constructed from 4 sequences derived from 4 individuals. Now such a graph is telling you what portions of the sequences are conserved across 4 individuals, right. So these are the conserved regions, whereas it is also letting you know what regions are varying across different individuals and how they are varying. Now we briefly touched upon how would we construct such graphs. So I briefly mentioned last time that if you are given some input genome sequences, then one way to construct such graphs would be to do multiple sequence alignment, because multiple sequence alignment would guide you towards what portions of the sequences are conserved across the sequences and what portions are varying in different sequences. So the idea was that once we know the conserved regions, that those would be collapsed into a single vertex, and the variable regions will be represented as alternative paths in the graph. So today the focus of this lecture will be on how to formalize this concept of multiple sequence alignment. At a high level, you can see that this is somewhat similar to pairwise sequence alignment, except that we are working with multiple sequences now. So we have to somehow extend the concept of pairwise sequence alignment to multiple sequences. So let's start with the introduction of multiple sequence alignment problem. As I said, this is a generalization of the concept of pairwise sequence alignment to multiple sequences. Let us see another example before we discuss a formal definition. So at the bottom I am showing you a multiple sequence alignment of this CTCF protein sequence coming from 8 organisms. So these organisms, as you can see on the bottom, these are from different species. So this includes mammals as well as zebrafish, and you can see how this sequence is conserved, how the multiple sequence alignment is telling you what portions of the sequence are conserved across different individuals and what portions of the sequence have mutations in them. Similarly, these mutations can be of type substitutions, or you may have some deletions or insertions in the sequences. And I hope you recollect, this is very similar to pairwise sequence alignment, except that we want to allow these character substitutions, insertion and deletions in multiple sequences together. So we will extend the definition that we had already looked at for pairwise sequence alignment. So this is a lecture slide from the previous lecture. We want to extend this definition now to multiple sequences. So let us see how to do that. So in case of multiple sequence alignment, we are given D sequences as input. I will be denoting them as T1, T2 up to TD, right. So this is the notation that I am using to denote D sequences, and the multiple sequence alignment of these D sequences is a D cross N matrix. That is, the matrix has D number of rows and it has N columns. Let me denote Mi star as the ith row of that matrix and m star j as the jth column of the matrix. So, this matrix, this D cross n matrix M is a multiple sequence alignment if for all I in 1 to D, that is for all rows from 1 to D, sequence T I must be a subsequence of row m I star, and the remaining n minus T I characters. So, basically the length of T I, you have a length of T I. So, T I will contribute its characters in the row, and the remaining n minus T I characters would be these dash characters which correspond to insert. So, this is to reflect insertion and deletions. So, as you can see, this is very similar to the pairwise sequence alignment definition, except that we are now working with D sequences, and instead of having instead of having columns with 2 values, now we have columns with D values. So, looking at one more example. So, consider these 3 sequence inputs T1, T2, T3. Then for them one possible multiple sequence alignment is shown here. So, this is having 3 rows corresponding to 3 sequences, and for this particular example we have 10 columns, right. So, because now sequence length of sequence T1 is in this case 9 and n equals to 10. So, therefore, first row will have 1 dash symbol. Similarly, the second row has 1 dash symbol and the third row has 3 dash symbols. So, overall this is a 3 cross 10 matrix. So, this is one example of a multiple sequence alignment. Now, the question comes, how do we score such an alignment, like how do we score a multiple sequence alignment, because we have to determine what a good alignment is versus what a bad multiple sequence alignment is. And once we are having once we have a scoring scheme, then we can formulate our problem to identify the multiple sequence alignment that has the highest score. So, once the scoring scheme is fixed, then we can look forward like how to compute a multiple sequence alignment efficiently, that is the alignment which achieves the highest score. So, let us understand these questions one by one. So, coming to the first question, how to score a multiple sequence alignment. So, let us look at 2 different scoring schemes for this. So, first as a first step, let us recall how we were scoring pairwise sequence alignment. So, remember that in a pairwise sequence alignment we were using this matrix format where we were saying, okay, let us consider match mismatch cost between A, C to C, C to A and so on, and once we had this match mismatch cost and insertion cost, we were just looking at individual columns and adding up the total cost or total score. So, for example, here G to G corresponds to score of 1. So, therefore, we were saying that this is +1, then further +1, then -1 and so on. So, we were looking at a scoring matrix and accordingly we were scoring a pairwise sequence alignment. Now, the question comes, how do we extend this concept to multiple sequence alignment, because it is not obvious now, because now in the case of multiple sequence alignment we have a matrix, so that each column instead of having 2 characters we have D values in every column. So, how do we extend this concept to a matrix, right. So, now instead of having two symbols in every row we have 3 symbols or D symbols in every row. So, because of these multiple symbols, it is not obvious on how do we score a multiple sequence alignment. So, let us look at the first scoring scheme, which is known as sum of pairs scoring model, in which we will directly extend what we have seen for pairwise sequence alignment to multiple sequences. So, here what we are saying is that let us consider any column J of the matrix M, what we will do is we will consider all d choose 2 pairs of characters in that column, and then we will apply the same scoring function that we were using for the pairwise sequence alignment. So, for example, if I consider the first column here, then I am saying that let us consider all pairs of these characters and apply this same scoring function that we were using for pairwise alignment. Similarly, if I consider this particular column, then the scoring function for this column would be SAC plus SCG plus SAG, because I am considering all pairs, and for this column you have pair AC, pair CG and pair AG. So, basically in other words, we are considering all d choose 2 pairs in every column, and we are summing the total score of all pairs of characters. If we aggregate this score over all columns, then we basically sum it over all n columns and we compute this value over the entire matrix. So, you can observe that in the outer summation is over j, that is ranging from 1 to n. So, this is for all columns, and the inner summation is considering the all pairs of rows in a particular column. So, here we are considering all pairs of rows I dash that are existing in a specific column j. So, now this is a very simple scoring function, right. So, this is easy to understand that we are directly extending what we used in pairwise sequence alignment. So, this function S that takes 2 symbols at a time, we are directly using that and considering all pairs of characters in a column. So, let us look at the pros and cons of this scoring scheme. The advantage of the sum of pairs scoring system is, first advantage is that it is simple, that it is a natural generalization from what we already saw in the pairwise alignments. However, one disadvantage is that it is not well motivated from an evolutionary point of view. So far we were just looking at the simplicity of definition. We are not, it is not clear whether this way of calculating the scores is consistent with the evolution of how these D sequences would have evolved. So, let me discuss this point with a specific example. So, let us consider these 7 sequences, and for a second let us focus on the last column of this multiple sequence alignment. Now, observe that you have 2 amino acids here. So, in these 7 protein sequences you have 2 amino acids N and D. Now, from an evolutionary point of view, it could have happened that at some point at the very top, maybe there was a mutation from N to D. So, let us say 3 sequences had N and 4 sequences had D, right. And further, that single mutation, that single substitution mutation led to 3 sequences having N and 4 sequences having D, right. So, that was a single mutation event in the evolutionary tree. But however, observe that the scoring system that we are considering, that is the sum of pairs scoring model, it is considering this substitution several times. So, for example, I will consider this N to D here, then I will consider N to D here again, then I will consider N to D here again, because there I am considering all 7 choose 2 pairs. So, therefore, I am considering this substitution multiple times instead of just one, because in the evolutionary tree this substitution could have happened only once. So, an obvious direction in which we can improve this scoring scheme may be to consider evolutionary tree of these 7 sequences as well when we are computing the score of a alignment. So, this brings us to another scoring scheme which is known as parsimony score. Here what we will do is again what we will try to do is we will define the score of our alignment based on the evolutionary tree of the sequences that are given to us. So, let us say that we are also given a tree which has D leaves, and this information is already known to us, let us say. So, now the question is, how do we use this tree to improve the scoring scheme? So, this should remind you of the problems that we looked at in our previous module on phylogenetic tree reconstruction. So, remember the small parsimony problem in a previous lecture, where we said that if you are given a tree in which the leaf labels are already known to you, then remember that we had identified a way in which the internal nodes can be labeled using the parsimony criteria. That is, we said that what is the, how can we, so we discussed an algorithm that where we identified the labels of internal nodes such that the parsimony cost is minimized over all possibilities. That is, we were identifying the labeling of internal vertices that minimizes the parsimony score. Now, we already know an algorithm to solve this problem from our previous lecture. The question is, how do we use this concept in the context of multiple sequence alignment? So, let us say, let us consider this example and let us consider the first column of this matrix. So, for this the labeling of the leaf nodes will be A, C, A, T, and let us say that we solve the small parsimony problem and identify the labels of internal nodes as well. Suppose they turn out to be A for all 3 internal nodes. Then what we can do is we can consider the branches of this tree and say what changes are happening across the branches of the tree. So, for example, there is a one substitution happening here A to C, then there is a one substitution happening A to T, and on the remaining branches we have A to A unchanged. So, based on this tree we can assign a score of 4 times S of AA, because there are 4 edges in this tree corresponding to AA vertices. Then there is one edge for corresponding to AC vertices, and then there is one edge corresponding to AT labeling. So, accordingly we can optimize our scoring scheme based on the knowledge of evolutionary tree. Observe that the sum of, so the previous model that we were discussing, the sum of pairs scoring model would have given a different cost to this column. So, that model would have considered all pairs of characters, right, and then it would have assigned the score, whereas in this scheme we are considering the evolutionary tree and based on that we are assigning the score. So far we have discussed 2 scoring schemes and we can see that the second one is better because it is more consistent with the evolutionary point of view. This brings us to the next step of the lecture, which is once we fix a scoring scheme, how do we compute the multiple sequence alignment? Because so far we have only seen the ways to score our alignment. Now let us fix a scoring system, and under that scoring system how do we compute an optimal alignment which achieves the highest score? Remember the dynamic programming methods that we have discussed for pairwise alignment. What we are going to do is we will extend those algorithms now for multiple sequences, and the basic trick for doing that is that remember the dynamic programming matrices that we were using for pairwise alignments were 2 dimensional. That is, if you were aligning 2 sequences A, B, then remember that one dimension we were keeping all the characters of A, and the other dimension we were keeping all the characters of B, and we were solving for each cell of this matrix using the recursion. For multiple sequence alignment, let us say that we have 3 sequences, we are going to make this 2 2D matrix into 3D matrix and develop a recursive algorithm accordingly. So, that is the overall intuition. Let us formalize this idea. So, let us say that, so let us fix for now d equals to 3. Let us consider a simpler scenario where the number of sequences given to us is 3. I will denote capital A ijk as the optimal alignment score of first sequence which is prefix of T1, second sequence which is prefix of T2 and third sequence which is prefix of T3. So, these prefixes are of length I, j and k respectively, and we will assume that we are using some fixed scoring scheme that could be the sum of pairs scoring scheme, parsimony scoring scheme or some other scoring scheme that exists in the literature. Now, extending the same idea that we used in a pairwise sequence alignment, the way we will develop the recursion is by looking at the last column of the matrix. So, remember that there are only finite ways in which the last column of the matrix can look. So, in the case of 3 sequences, what are different possibilities? So, let me consider xi as the ith sequence of T1, similarly yj as the jth character of T2, and let us say zk is the kth character of third sequence. Then what are the possible ways in which the last column of the matrix can look? Well, in the last matrix it may be possible that you have xi, but maybe in the other 2 sequences you have a dash character. The other possibility is that you have a yj here and the third sequence has a dash character and so on. And you can see that there are in total 2 raised to power 3 minus 1 possibilities, right. So, not counting the, so this is not allowed because we cannot have a, it does not make sense to have a dash symbol in the entire column. So, leaving this case aside, you have 2 raised to power 3 minus 1 possibilities. Accordingly, we can develop the recursion. So, in that case ai j k would be maximum of all the possibilities, that is maybe the last column has all 3 symbols, then in that case we will recurse using the previous value ai minus 1, j minus 1, k minus 1 because the 3 symbols have been used in the last column. Similarly, the other possibility is sxi dash dash, in which case we would use ai minus 1 comma j comma k. So, as you can see this is extending the pairwise alignment concept, except that now we have to consider multiple cases because the column length is longer now. So, then basically we can solve this matrix. So, this is a 3-dimensional matrix now, and we can follow a ordering in which how to compute these elements of the 3D matrix, and we can solve those elements one by one. Now, as far as runtime of this algorithm is concerned, then for that we will have to look at how many values are we solving, that is how many sub problems are we solving, and how much time is it taking to solve each sub problem. It is slightly more, like we have to be slightly more careful for multiple sequence alignment compared to the pairwise sequence alignment, because for pairwise sequence alignment each cell was depending on fixed 3 neighboring cells, whereas now this number depends on the how many sequences are we considering, because this is still d equals to 3 scenario. For d equals to 4 we will be moving to 4-dimensional matrix, for d equals to 5 that would be a 5-dimensional matrix and so on. So, let us analyze the runtime. For 3 input sequences, observe that there are T1 times, T2 times, T3 times sub problems, that is the product of the sequence lengths. So, this is the size of the matrix, because each dimension of the matrix so in the matrix that we have, each dimension of the matrix is corresponding to one sequence. So accordingly we have these many sub problems, and solving each of which takes 2 raised to power 3 time, because observe that we have to consider max over all these possibilities. What about the general scenario where you have D sequences? Well, in that case we will be working with D dimensional matrix. So in that sense we will be solving T1 times, T2 times, T3 times up to TD sub problems. And how much time does each sub problem take? That is, how much time are we spending to compute the recursion that we have? Well, observe that first of all there are two components of the computation, that is, we have to solve for these value S, right. So we have to find out the score of that column, and we have to calculate that score 2 raised to power D times, because we have to consider all 2 raised to power D possibilities. So how much time does calculation of score take? That is, how much time do we have to spend to compute this S function? Well, if you consider the sum of pairs cost model, remember that we have to consider all D choose 2 pairs. So therefore it is taking order D square time to calculate the score function S, and we have to calculate this score function 2 raised to power D times, because that we have to consider all 2 raised to power D possibilities for filling that value when we are computing the max function. So considering this algorithm, basically the runtime of this algorithm will be the sequence length. So that is the time, that is the number of problems that we are solving. So this is D times D square times 2 raised to power D. Sorry, this is wrong. So let me correct this. So this will be 2 T1 T2 TD, so times D square plus 2D. Right. So that is, we are spending D square time to compute this cost. So and we have to compute max over all 2 raised to power D possibilities. So this should be a product here, because we are computing this score D square times. So what would be the score if we use parsimony score? So what would be the run time if we use parsimony score? This I will leave this as an exercise for you. What you will notice is that if you work this out and if you recollect the time that we were spending to solve the small parsimony problem, you will find that basically this factor will reduce, because instead of considering all pairs of scores, all pairs of characters, now you have to consider you have to solve the small parsimony problem and consider the edges of the tree into account. So this, when you solve this exercise, you will find that this time should reduce, but the other factors will remain same, because the recursion will remain same, and still we have these many sub problems to solve. Okay. So I want to make some remarks about this problem. The first step is that observe that the time complexity that we have for this problem is extremely slow. So that is that it is spending as much time as the product of lengths of those sequences times D square times a factor which is exponential in D. So if we want to scale these to many, like large number of sequences, then you can imagine that this algorithm will become very, very slow. But unfortunately it turns out that using complexity theory we can prove that significantly faster algorithms are unlikely. So this problem belongs to a class of hard problems, and because of that it is unlikely that faster algorithms will exist. If you want to read more about the proof, you can refer to this paper, which does this which uses this complexity theory to prove that the multiple sequence alignment belongs to the class of hard problems. The second remark I want to make is that in practice, because computing exact multiple sequence alignment is often unfeasible for large number of sequences, most implementations would use ad hoc heuristics, and among those heuristics, progressive alignment is the most common heuristic. So what do I mean by progressive alignment? Well, as the word suggests, we will build the multiple sequence alignment progressively. In this scenario, in how this works is basically we will use a phylogenetic guide tree, right. So we will have a tree that will guide us on how to do the progressive algorithm, and suppose we have a tree. So let us say that we have a tree T1, T2, T3, T4. So we will progressively combine the neighbors. We will do a pairwise sequence alignment of the neighbors. Similarly, I will do a pairwise sequence alignment of the neighbors T3 and T4, and then I will go in the bottom up fashion and I will do a alignment of the multiple sequence alignment of these two sequences. So basically, as we go in a bottom up manner, we will combine these multiple alignments of size 1, 2 and so on until we reach the root node of the tree. So in other words, we are starting from the neighboring leaves of the phylogenetic tree or the guide tree, and we are working our way in a bottom up fashion. We are combining these multiple alignments as we go up. So as you can see, as we are going up, we will be the number of the column size will increase, because that is how we are adding these sequences in a progressive manner. This idea will become more clear if we look at an example. So let us say that these are the 4 sequences given to us, and at the bottom I am showing you a guide tree. As a first step, what I am going to do is I am going to compute pairwise alignments of T1 and T2, and similarly I will compute a pairwise alignment of T3 and T4. So we have a pairwise alignment here and we have a pairwise alignment here. So this step is taken care of. Now as I go up in the tree, I would want to combine these two multiple sequence alignments. Now the question comes, okay, we know how to align two sequences. How do we combine two multiple sequence alignments? Because this is first multiple sequence alignment, this is the second multiple sequence alignment. How do I combine these two alignments together? Well, the trick for that is basically what we can do is we can assume each column as a symbol. So for example, I can treat this as a symbol GA, and I can treat this as a particular symbol -G, and I can repeat the pairwise sequence alignment algorithm. So in the pairwise alignment algorithm, I will consider this as a character and this as a single character, and the cost of aligning these columns GA and -G will be governed by what is the cost of aligning the entire column GA -G, that is the concatenation of both columns. So that will be the cost of aligning these two columns. So following this idea, what we can do is basically we can now combine these two alignments into a larger multiple sequence alignment, and we can continue this up. For example, if there were 5 more sequences T5, T6, T7, T8, then I can repeat this process on this part as well, and now at this part I will have to combine two multiple sequence alignments of size 4 each. So again I will repeat this idea, and subsequently I will combine this further. So you can see this is why this is called as progressive alignment, because we are progressively building the multiple sequence alignment, right. So that is a commonly used heuristic in most implementations, and this is much faster than the exact algorithm. However, this heuristic will not guarantee that it is returning an optimal output, but in practice it often returns outputs which are very close to optimal. The final remark I want to make before we close this lecture is that multiple sequence alignment is a fundamental concept in bioinformatics and it is used for many applications. Now, one such important application is the protein structure prediction. I assume that most of you would have heard of this AlphaFold breakthrough in the scientific community, where extremely good results were achieved for protein structure prediction problem. Now, what I want to emphasize is that in this model architecture that was used by the authors, they use multiple sequence alignment as a feature in the model. So, if you are given if you give the model an input sequence, it will do a database search to identify closely related sequences and it will do a multiple sequence alignment of those sequences, and that multiple sequence alignment will be fed to the model during both training as well as inference stages. Okay, so now you can ask, why would a multiple sequence alignment of protein sequences help during protein structure prediction? Well, the reason for that is that from the multiple sequence alignment, the model can see what characters are conserved well across multiple sequences, and from there it can identify if the characters are well conserved, then those must be structurally and functionally important sites. Similarly, the other factor that you can think of is that if there are two characters which are far apart in the multiple sequence alignment, but let us say that they are evolving together, that is, mutation in one increases the chance of mutation in the second sequence, in the second character. Even though they are far apart in the 2D structure, they may be spatially close in the 3D structure, because if they are co-evolving together, then it is likely that they are spatially close in the 3D structure. So, these kind of insights are useful for the deep learning model, and that is how multiple sequence alignments play an important role for protein structure prediction as well. So, this brings us to the summary of the lecture. So, in this lecture we looked at a fundamental problem for multiple sequence alignment. We discussed it in the context of pan-genome graph construction, where you may have a collection of sequences for which you want to build a pan-genome graph, and multiple sequence alignments can be used as a guiding mechanism to build that graph. But multiple sequence alignment has many diverse applications, including protein structure prediction, understanding evolution of sequences and so on. So, I hope by now you understand how multiple sequence alignment works, how is it defined, and what are the algorithms that we use for solving this problem. Thank you.